Okay, greetings students. I am going to try to give you a quick primer on DNA structure so that you're going to be better prepared for transcription and translation. The obligatory cartoon is reminding us of our base pairing designs. So that is adenine A bonds with thymine, guanine G bonds with cytosine. So G and A would not bond. We've already seen this, right? Here's our phosphate, this is our pentose sugar, and there's our nitrogenous base. So these are the three parts. And again, this is what it looks like. This happens to be the nitrogenous base adenine. So this is the part that we're going to be looking at that's going to be changing. Note here as well on the second carbon, there's no oxygen. So this is DNA, deoxyribose, not RNA. That's the same thing that we already talked about in class. That's ribose. All right. Now, when you have free-floating nucleotides, this is what they look like. And there are three phosphates, 1, 2, 3. And later on in this class, we'll spend a lot of time looking at this, but I've already actually talked about it a little bit. I said all energy is in the form of ATP, and this is adenine triphosphate, so that's 3 nucleotides. And so you see that that is actually a nitrogenous base. This is what it looks like in the rung of the DNA ladder, but this is what it looks like as energy. So the good part about this is you're already learning some of these basic molecules, and the other good part about this is if we have time, it helps you understand how DNA gets the energy in order to be replicated. And we've talked about this. This is just, again, dehydration synthesis, right? You have the H here, the OH here. Those things come together. Water is created. And then you get a bond between the 3' carbon here, or the oxygen that's off the 3' carbon here, and the oxygen off the phosphate here, which comes off the 5' carbon of the ribose sugar. So this is how you link two nitrogenous bases together. This image, again, is meant to emphasize that DNA has a particular shape. So it has a 5' end, because if we have a first carbon, second carbon, third carbon, fourth carbon, fifth carbon. So if that's on that fifth carbon, that's the 5' end. This side, you should see, this goes first carbon, second carbon, third carbon. So this ends up being the 3' end over on this side. So DNA has a particular direction, a 3' and a 5' direction. So this slide shows you the 4 nucleotides, adenine, guanine, cytosine, and thymine. It also classifies those nucleotides into 2 groups. So you have purines and pyrimidines. What you should be able to see is that purines are going to have 2 rings, 1, 2, 1, 2, whereas pyrimidines are going to be smaller, and they're only going to have 1 ring each. So adenine and guanine are the purines, and cytosine and thymine are the pyrimidines. And one way that you can remember this is this little quote. Good, the G in good stands for guanine, angels, and the A in angels stands for adenine, are so pure, they are purines, guanine and adenine are purines, they have 2 halos. Halos are the rings. I probably misspelled the word halos. So I hope that helps with that. This is just another specific, like more specific focus of what the different nitrogenous bases look like, so you can see them. So the one thing I want to point out, especially as we start looking at RNA, you may remember that thymine is replaced by uracil in RNA, and the only difference between the thymine and the uracil in this RNA is right here. You have this extra carbon. So here in thymine you have an extra carbon. Here in uracil it just bonds to hydrogen there. So that's the difference between the thymine and the uracil. This is just showing you the fact that DNA goes from 5' to 3' on one side and 3' to 5' on the other. So what I've done is I actually just took this image and I flipped it around. So you can see the 5' end's on that side, the 3' end is on this side, and if you flip it upside down, that's the 5' end, that's the 3' end. This process is called, the fact that they're running, the two strands of DNA are running in opposite directions. It's called being anti-parallel. This is what it looks like. Just another image here, 3', 5', 3', and 5' there. And you can see that you see the oxygens on these. So these sugars are pointed in this direction. And then the oxygens on this side are pointed down. So in fact, these two strands are flipped. Another thing that you may notice is that if you have adenine and thymine, you only have two bonds. These bonds are hydrogen bonds. But if you have cytosine and guanine, you have 3 bonds. So here's guanine, I'll put a G here. And cytosine, you have 3 bonds there. That ends up being important when we do transcription as well. Another thing that I want you to see is that every time you have a nitrogenous base that only has 1 ring, it's always bonded with a nitrogenous base that has 2 rings. So the total number of nitrogenous bases is going to be 3. And this is helpful because otherwise your DNA would be like fat sometimes. So let's say if you had a 2 bonded with a 2, or super skinny, like if you had a 1 bonded with a 1, right? And so by always having 3 nitrogenous bases bonded together, you have an equal width of your DNA. And as we've talked about before, you can see in here, these are all hydrogen, carbon, nitrogen bonds, so these are all non-polar. So they are on the inside of the DNA molecule. You have obviously these charged sections on the phosphate that are over here, and you have oxygen-hydrogen bonds on the outside here, and so this is polar. So you have a polar region to DNA, polar region to DNA. This just discusses the differences between DNA and RNA. This is a slide you've seen already. So the purpose of this slide is to show you that because you know what adenine bonds with and you know what guanine bonds with, you only actually need half of the DNA molecule in order to be able to predict the other half of the DNA molecule. So if you look over here and you can see A here, A is going to bond with T, T is going to bond with A, And so you can just keep going all the way down and create the complementary side of the DNA molecule as you do this. And the other thing that I'm moving you towards is this question about when you're splitting DNA apart, would it be more difficult to split DNA when you have a bunch of guanine and cytosines, or if you have a bunch of adenine and thymines? Where is it going to be easier to split apart? You see that this has got 3 hydrogen bonds. This has 2 hydrogen bonds. So hopefully you can see that this is going to be the easier side to split apart. And then finally, I want to stop with this idea of histones. So since DNA or human DNA has 3 times 10 to the 9th base pairs, and only takes it 75,000 base pairs to stretch across the nucleus, you could stretch across the nucleus with human DNA 400,000 times. And so in order to deal with this issue, what happens is DNA is compressed very, very specifically. So this is the double helix down here that you're used to seeing. Now, this is wrapped around this thing called a histone, and I'll be talking a lot about histones. Doing that gives you a nucleosome, and you actually get 147 base pairs wrapped around a single histone. So you get a fair amount of concentration that way. And then 8 of these nucleosomes will wrap around to make this kind of thicker thread. And then that thicker thread itself will become condensed and get coiled. And then that coil itself will become coiled again. So really you have 1, 2, 3, 4, 4 levels of concentration in order to get the DNA into this small area. Now this actually is during replication. So it's very rarely like this, but DNA is quite often in these 2 shapes here when it's in the cell doing its job. All right, so you can think about the things you understand or you're wondering about that are going to help you learn, but this is the fast overview of DNA replication.